First of all, of course, I would also like to thank the organizers for inviting me and giving me the opportunity to talk to you today. It's a very exciting meeting and the program is fantastic. And I know I'm in an anatomy session, but I wanted to switch gears a little bit and talk about functional anatomy of in particular, the rodent prefrontal cortex, and in the context of cognitive control, so the control of behavior. Of course, so our society relies heavily on the control of behavior, so inhibitory control of behavior. We cannot live in a busy city like this, for instance, if everybody just follows their impulses. So control of inhibition is control of behavior is very important, inhibitory control, and we spend a lot of time and efforts in training humans from early ages on to control their behavior, to ask if they can go to the bathroom, for instance, before they actually go to the bathroom. So, in the human brain, this control of inhibition is organized by activity in prefrontal cortex, striatum, and thalamus. In this particular task, where in this study, people were asked to wait for a light to signal that they can start moving their hands, and then if another light would come on a little bit later, they would have to stop one of the two hands and thereby control their behavior, their output. And while being in the scanner, the activity of striatum, orbital frontal cortex, thalamus, and inferior frontal cortex were recorded. But of course, these were recorded in fMRI, so the temporal resolution is not very high. It's a correlative observation, of course, and what we want to know, what I want to talk to you today about, is how is this activity actually organized to orchestrate this cognitive control of behavior. And for that, to address this and to understand in a causal manner how the prefrontal cortex, striatum, and the projections to striatum and thalamus are involved, I want to move to rodents. And what we did is we studied the role of specific outputs from the prefrontal cortex to different parts of the striatum and to different parts of the medial dorsal thalamus, which is of course known to have heavy connections with the prefrontal cortex. So from early anatomical studies, we already know that there are specific output neurons in the prefrontal cortex. These are not these can also be in orbitofrontal and the lateral frontal areas of the rodent, but in the rat there's a heavy connectivity with the medial prefrontal cortex between the medial dorsal thalamus, ventral striatum, and dorsal striatum, as you can see here. And these neurons are intermingled, but there are gradients. So you see that the purple neurons are the neurons that project to the medial dorsal thalamus. You see that the red neurons are projecting to the dorsal striatum, and the blue neurons are projecting to the ventral striatum. There's a gradient here, and the medial dorsal thalamic projecting neurons seem to be in the more deeper layers of the medial prefrontal cortex of the rodent. So we wanted to address the question how the function of these output neurons is involved in the inhibitory control of behavior. But first, of course, the medial dorsal thalamus is not a homogeneous structure. It also has different parts. It has a lateral part, central part, and a medial part. And so first what we did is we did a little bit of a tracing study as well to identify the projections and the locations of the prefrontal neurons that project to the different parts of the medial dorsal thalamus. So we injected retrobeats, and we found that there's a distribution of projection neurons to the lateral part of the medial dorsal thalamus that are located in the deeper layers of the dorsal medial prefrontal cortex. We did the same for the medial part, and found that the projecting neurons are mostly located on the ventral side of the medial prefrontal cortex. For the striatum, the dorsal striatum, that was similar to what Gabbett found. So we find that there's projection neurons in the more superficial layers on the dorsal side that project to the dorsal medial striatum. And then for the ventral medial striatum, we find that they are located also on the ventral part, on the more superficial layers of the medial prefrontal cortex. And now the question, of course, is, so then we have this distribution of all these cells. And first of all, we wanted to know, so you have in the ventral part, you have neurons that project either to the striatum in the more superficial layers, or to the medial dorsal thalamus and then the medial part. And do some of these neurons actually project to both of these areas, or are these separate neurons that project to these anatomical output targets? And the same holds true for the dorsal part. So you have these neurons that lie intermingled, that project to the dorsal striatum and the lateral part, the medial dorsal thalamus, but are these the same neurons, or do they segregate? So we took a combinatorial viral approach, injected FLIP and CRE-dependent viruses in the different parts, in the dorsal striatum and in the lateral part of the medial dorsal thalamus, and then looked in the dorsal medial prefrontal cortex whether these targets would segregate or not by separating the EYFP and M-cherry colors. And we also stained for GAT67 so that we know whether they were inhibitory neurons or not. And what we find is basically that there's almost 100% separation of projections. So only 0.8% of the cells project to have M-cherry and EYFP. So most likely these populations are purely independent, almost purely independent. And the same holds true for the ventral side. So where we inject M-cherry and EYFP dependent on CRE or FLIP, and find that these projection neurons are projecting either to the striatum or to the medial dorsal thalamus. There's only a very small percentage that has both colors. So these are separate output populations. So we come to an anatomical description of the projections like this. So the dorsal part of the medial prefrontal cortex projects to the lateral part of the medial dorsal thalamus, the ventral part to the medial part, and for the striatum, the ventral part projects to the ventral medial striatum, whereas the dorsal part projects to the dorsal medial striatum. Now, of course, so this is the anatomy, but what are these projections doing for inhibitory control? So to measure inhibitory control in rodents, you can do different kinds of tasks. We chose for a task a home cage-based task where the animals can choose to participate in this task themselves. So their home cage is connected to the operant chamber via a tunnel. The tunnel opens only for a couple of hours at the start of the dark phase, because that's when they become active, and that's when they can do a lot of trials, and they can do the task as much as they want during those 2.5 hours, and earn their food by doing the task. So what is the task like? So the animals have to observe a wall with 5 holes in it. After some time, a light will come on for a certain amount of time, and then they have some time to make a response, so stick their nose into the hole that was lit, and thereby earn a food reward that is delivered at the other end of the cage. So after that they can start a next trial and do that as much as they want during the 2.5 hours. So they can make a correct response and earn the food reward. They can make an incorrect response, or they can omit, so they not make a response. We think that these parameters are related to attention. How well is the animal attending the holes? But also the animal, since they want to earn their food rewards, they can also make premature responses, so stick their nose into the hole before the light has come on, and this is related to impulsivity. And during this delay period, the animals, of course, have to withhold from responding, have to inhibit their behavior, and this is the time that we are interested in for these outputs. So what are these outputs doing during the, just a little bit of behavior. So when you do this, the classical form of the 5-choice serial reaction time task, then it takes animals about 3 weeks to learn the task. But if you give them unlimited access to the task from their home cage, they can learn it in a couple of days and become experts within a week, even if they have time-restricted access for 2.5 hours in the dark phase. So to figure out what these projection neurons are doing, we expressed dread receptors in the projections using a combinatorial viral approach again. So labeling or expressing inhibitory dread receptors in either the projections to the medial dorsal thalamus, the lateral part, or the medial part, or the dorsal striatum, or the ventral striatum. And these are the locations of the labeled neurons. And then we did this task, but the animals were trained on a fixed time schedule in this task. But then when they were tested, with we changed the delay period. We made the delay period unpredictable, so that it would randomly would be either 5 seconds, 7.5 seconds, or 12.5 seconds that the animal would have to withhold responding before the light comes on, so that they were really, how you say it, challenged on their inhibitory control. And what we typically observe is that when you do this, then animals start to increase premature responses, especially on the longer delay times. It's just because they are less able to wait that long before making a response. So they become more, the inhibitory control becomes less with these long delayed trials. Okay, so and then what we did is we applied different concentrations of CNO in different sessions, and what I plot here is the change in premature responses relative to no CNOs, relative to control. So what you see is the different concentrations, and then in the different projection neurons to the medial dorsal thalamus, the lateral part, and the medial dorsal thalamus in blue, the medial dorsal cell medial part in green, and then the EYFP control in gray. And we plot the difference. So you see that with the 5 seconds delay, there's no change of the inhibiting these specific projections, either to the lateral part or to the medial part. When you look at the larger delay of 7.5 seconds, you already start to see an improvement, a reduction of the premature responses when these projections to the lateral part of the medial dorsal thalamus are inhibited, and when you look at the longest delay period, where it's most difficult for the animals to show this inhibitory control, you see that in particular the projections to the lateral part of the medial dorsal thalamus, when you inhibit those, then the inhibitory control becomes better. But when we inhibited the projections to the medial part of the thalamus, the premature responses went up, and that showed a reduction of inhibitory control. At the same time, we can also look at the attention parameters. We saw that when we inhibit the projections to the lateral part of the medial dorsal thalamus, we saw an increase in the omissions. So the animals more often started to not respond when the cue light was come on. For the other parameters were not changed. So what we conclude from this is that normally this projection dorsal prefrontal cortex, medial prefrontal cortex to the lateral part of the thalamus is a sort of a go signal. When you inhibit this, then the animal will be better able to inhibit its go behavior, whereas the other projection from the ventral side of the medial prefrontal cortex to the medial part is more of a stop, has more of a stop role in this behavior. If you inhibit that, then there's more premature responses, so the animal is less able to inhibit its behavior, responding behavior. We did the same for the striatal projections, and what we observe is that when you inhibit the projection from the medial prefrontal cortex to the dorsal striatum, we see an increase in the premature responses, which means a loss of inhibitory control. And the projections to the ventral striatum, inhibition of those. This is the omission, sorry. The ventral striatum, that's the purple ones, that didn't change the premature responses, and also the omissions were not changed when we inhibit both projections. Suggesting that this projection from the dorsal prefrontal cortex to the dorsal striatum is also a sort of has a role of stopping or inhibiting the behavior. When we inhibit that projection, then the animal shows more premature responses, less cognitive control. So when are these projection neurons active? So are they actually, what kind of activity do they show during this behavior? So we took two approaches. We did either recorded the activity of these projections by recording GCamp activity in the cell bodies of the prefrontal cortex neurons that project to the different targets. Or we used wireless optotact electrophysiology. So we expressed channelrhodopsin in the projections, had a multi-electrode array in the prefrontal cortex, recorded action potentials or unit activity and tested with the turning on the laser whether we were recording from neurons that project to a specific target, in this case to the dorsal medial striatum. So first to show you the results from these wireless, and it was wireless so that the animal could just do freely moving behavior while we were recording the different units. So what we find is that during that many of the neurons, the optotag neurons, they increase their firing during the delay period, so during the inhibitory control period. And when we increase the duration of the delay period randomly, then you see that these neurons have their activity at the same time as the delay period. So as long as the animal has to wait and before responding, the neurons are active, and then at the moment they respond, then the activity drops and they collect their reward. We also find that the population of neurons is inhibited selectively during that period. So some of the neurons reduce their activity during that period, and there are also neurons that actually don't show any change in firing activity, suggesting that they may be involved in some other behavior. So when we look at the different trial outcomes, so animals can make correct responses or incorrect responses or premature responses, then what we find is that in particular during the premature responses, so when the animal does not successfully inhibit its behavior, the activity of the neurons is different. So it shows reduced activity and it goes down more rapidly. And this holds true for all the delay periods, but especially on the long delays, of course, there are more premature responses, so we had more examples of those. And also with the neurons that are inhibited during the delay period, they show much less of the inhibition during this, when they make a premature response. And that's quantified over there. But at the same time, what we also observe is in the behavior that when an animal is going to make a premature response, that it is more eager, or it turns around more rapidly, and it crosses this bar. We were filming the animals crossing this bar more rapidly. So this is the behavior. So this is the time between the start when it sticks its nose into the food magazine to start the trial. The time difference between the start and the turning around to the cue, to orient itself to the cue lights. You see that in trials where the animal makes a premature response, this time is shorter. So it's as if the animal is more hasty, is more eager to start the next trial, earn a new reward. And when we look at the activity of the neurons that project from the prefrontal cortex to the dorsal striatum, we also find that their activity goes up earlier than with correct trials. So this is plotted here in the delta, the change in the spiking density of these neurons during this trial. So to conclude that, so what we observe is that the difference between correct trials where the animal is correctly inhibiting its behavior compared to the premature trials when the animal is not able to withhold its behavior, the prefrontal neurons start to be active more, that project to the dorsal striatum are active earlier on and show less activity than in correct trials. So what are the other? When are the other projection neurons active? So for this we used the fiber photometry. Of course, it has a much lower temporal resolution, and also, of course, we are looking at a mixed signal between neurons that increase their behavior, their activity, and neurons that decrease activity. But anyway, we had GCAMs in the different projections. From to the medial dorsal thalamus, lateral part, medial part, to the dorsal striatum and the ventral striatum. And to make a long story short, what we can see differences in the different trial outcomes, especially when you look at differences between, for instance, correct and premature. We find that these projections to the lateral medial dorsal thalamus show a difference in the amount of activity and the amount of fluorescence signal that we record in the fiber photometry. But surprisingly, of course, what we found earlier with the behavior is that one of those projections is more of a go signal, and the other is more of a stop signal. But we find that both populations actually show an increase in fluorescence during recordings. But there are differences between the different trial outcomes, suggesting that indeed also the activity of these neurons is different between correct responses and premature responses. Oh, yeah, and importantly, the projections to the ventral striatum, they didn't show any change in fluorescence, much of a change in fluorescence during the delay period in this task. So it's only those other 3 projections that show changes in activity, task-related changes in activity. All right, so to conclude, what I've shown you today is that there are specific projections from the medial prefrontal cortex to the different parts of the thalamus, different parts of the striatum. That these have, they are anatomically segregated between the dorsal and the medial side, but also between deep and superficial layers of the medial prefrontal cortex. These projections allow for a bidirectional command of cognitive control of behavior. So some of these projections, especially to the lateral part of the medial dorsal thalamus, tell the animal basically to engage, to go, and whereas other projections here in this the ventral side to the medial part of the MDT is more of a stop signal, as same as the projections to the dorsal striatum from the dorsal medial prefrontal cortex, and thereby you get several axes of bidirectional control. You get one between the dorsal and the ventral. PFC, the dorsal and the ventral PFC, a stop and a go, bidirectional control, but also between the deep and the superficial layers of the dorsal medial prefrontal cortex. So they are active during the delay period. So when the animal is actively inhibiting its from responding, these different projections are all active, and they encode the behavioral trial outcome. So when the animal is not able to withhold its responding, these projections show less activity. And that's basically what I wanted to show you. So I have to thank a lot of people, of course. Huib, Bastiaan, and Sybren did all the experiments, the behavioral experiments, and the various physiological experiments. Our colleagues from the other department who developed first this home cage-based behavior for mice, we adopted it for rats, and our colleagues from the medical center. And the funders, of course, and I want to thank you for your attention.